Hello, FBlock. This is my cell communication project on SARS-CoV-2 spike protein epitopes, better known as COVID-19. So before we get to the ligand, we have to talk about how the ligand is actually going to be made. And this is going to start with the virus cell infecting the host cell either through receptor binding to the ACE2 receptor or it's going to fuse with the membrane. Now, as this happens, it's going to come into the cell and it's going to go through something called uncoating. And when this uncoating happens, its RNA is going to be released into the cell, going to be processed, and then it's going to go into the rough endoplasmic reticulum and it's going to make more virus cells. Sometimes when these viruses are being made, instead of making it to the virus cell, they will be intercepted before. One of these proteins that are made is spike proteins, which is the best well researched, and it's the one I picked. When the spike protein doesn't make it to the virus, it is instead going to be broken down by something called a proteasome, as we can see here. Now, this proteasome is going to come and it's going to break it down into something called viral peptides. These viral peptides are going to go and they're going to bind to the MHC1 complex. As this happens, it's now become an MHC1P complex. And this is the point where these peptides are going to be called epitopes. Now, this epitope MHC1 complex is going to come and it's going to move to the membrane. On the membrane, it's going to attract something called TCR, which is a non-catalytic tyrosine receptor on CD8 plus T cells, which are very similar to T cells. And what's going to happen is this TCR is going to bind through juxtacrine signaling. And as this TCR cell binds, as you can see here, something else called a co-receptor, which is CD8, is going to bind as well. When this binds, it's going to activate the LCK kinase, which is going to go and activate ZAP70. Now, when ZAP70 is activated, it's going to come and it's going to get recruited by the LAT signal zone. When ZAP70 joins this protein complex, it becomes the LAT multiprotein. Now, this LAT multiprotein group is going to come and it's going to activate RAS, which is a G protein. This G protein RAS is going to come and it's going to activate RAF. And this RAF is going to start a phosphorylation cascade from RAF to MEK to ERK. Now, this ERK is going to come, it's going to phosphorylate our transcription factor AP1. AP1 is going to come and it's going to enter the nucleus and we're going to get to the response. Now, the response is the gene expression of three different things: granzymes, the little red things, perforins, and then cytokines such as TNFA. These cytokines, which I'm not going into detail, will basically promote inflammation and they're going to cause more T cells to divide, meaning a better immune response. Back to the granzymes. Here we can see a granzyme, and it's going to enter through these perforins, which create pores in the cell membrane of the host cell. These granzymes are going to enter the cell, and they're going to activate something called caspases, as you can see here. And finally, these caspases are going to come, and they're going to activate apoptosis. When this apoptosis happens, the infected cell will be killed, and the infection will be stopped. That was a lot. The end. Thank you.